sterilization. STERIS Tech Talks are a series of webinars covering subjects relating to gas and radiation sterilization processing and the laboratory testing and validation services which support these processes. My name is Ashley Marute, and I'm the Associate Product Manager for STERIS Applied Sterilization Technologies. I'll be the host for today's event. Our presenter today is Jason Rogers, a Principal Scientist at STERIS AST. Jason graduated from the University of Minnesota Twin Cities with a bachelor's degree in biology. Jason has over 15 years of experience in the medical device industry and contract laboratory testing. He also represents STERIS on several AMI committees, including microbial methods and sterility assurance. All attendees are on mute for the presentation. However, we would like to encourage everyone to submit questions using the questions function on the GoToWebinar control panel. Questions will be answered following the presentation. Today's presentation will be recorded and uploaded to our STERIS Applied Sterilization Technologies YouTube channel. Please note that continuing education credits are not provided as part of this webinar. And now over to Jason to begin the presentation.
Good morning, and thank you for joining us for today's Tech Talk for testing and validation for gas sterilization. The key outcomes for today's webinar are to cover the testing processes that may be used in the validation of gaseous sterilization modalities, primarily ethylene oxide. We will also review the test method validation requirements for tests as required by the applicable standards guidance. Additionally, we will discuss the concept of sterility assurance level and residue testing. We will start by reviewing some key terms such as bioburden, which is the number of viable microorganisms on or in a product or sterile barrier system. Bioburden is measured in CFU, colony forming units, which is an estimate of the number of viable bacterial and or fungal cells in a given sample. Tests of sterility are presence or absence tests performed on the product or its components in the execution of a validation. Tests of sterility of the product are performed after low exposures to the sterilant in order to show comparative resistance to a biological indicator organism. Sterility assurance level is the probability of a non-sterile item. Bioburden is comprised of bacteria and fungi. Fungi include yeast and molds. Fungi are eukaryotic cells with a defined nucleus, chromosomes, and lipid-bound organelles, where various cell functions take place, such as storage and metabolic activities. Here is an image of yeast cells. Notice the small cells coming from the larger cells. This is a form of reproduction called budding. This is an image of a penicillium mold species. Each circular structure is a fungal spore containing genetic material allowing for the proliferation of the mold. Each spore on a device surface would need to be inactivated to achieve sterility. Bacteria are prokaryotic cells that do not have a defined nucleus, chromosome, or lipid-bound organelles. The cell functions are carried out in the cytoplasm of the cell. Bacteria have many different shaped cells. Here we see a cocci or spherical shaped cell. Staphylococcus would be an example of a cocci shaped cell. Some bacteria are bacilli or rod shaped cells. Some rod-shaped bacteria can also form endospores. The endospores have a spore coat that forms to protect the genetic material in unfavorable environments, protecting it from environmental conditions, similar to a plant seed, until more favorable conditions occur and the spore can germinate. Infectious particles, such as viruses and prions, may be of concern for tissue-based products. Additional information for testing of infectious particles can be found in the ISO 22442 series. When we discuss bioburden of medical devices, it is usually referring to bacteria and fungi. Infectious particles may also be a concern for devices that are reused within a hospital. The considerations for cleaning, disinfection, and sterilization within the hospital are covered in ISO 17664 and ISO 15883. In general, for medical device cleanroom manufacturing, personnel are sources of gram-positive cocci, such as Staphylococcus and Micrococcus. Soil is a common source of bacterial endospores and fungi. Water and water systems are sources of gram-negative bacteria, such as Pseudomonas species. Gram-negative bacteria are the source of bacterial endotoxins. Damp conditions are favorable to fungi and gram-negative bacteria. Outside air contains soil organisms and fungi, and cleanroom air typically contains organisms from personnel. Now that we discussed what bioburden is, we can review the manner by which we culture the organisms. The bioburden tells us what our contamination level is, and can also give us detail about the resistance to sterilization. The lower and less resistant the bioburden of the product is going into the sterilization process, the greater the chance of success in the sterilization validation. In a bioburden test, we start by preparing the sample for extraction. Using sterile tools, a testing technician may cut up, disassemble, or flush a device. The device is then placed into a sterile extraction container, such as a bag or jar. A sterile pH buffered solution, usually containing a surfactant, is used to rinse the device and extract bacteria and fungi from the surfaces of the device. The method of extraction could include shaking, sonication, vortexing, stomaching, or a combination of methods. After extraction, the liquid is filtered and plated onto agar media, or it may be plated by mixing the liquid directly with molten agar, referred to as pour plating, if filtration is not an option due to the nature of the test sample, whether it is fibrous, powder, or gel. Other culturing methods such as MPN, most probable number, contact plating, and swab culturing may also be options. We perform testing on certain media types or test conditions to better categorize and understand the bioburden. We can also perform organism identification on recovered colonies to further understand potential sources of contamination. Whichever culturing technique is selected, the method is validated to measure the efficiency of the method and correct the result with the use of a correction factor. There are two methods identified in ISO 11737-1 for the determination of the correction factor. Both methods have advantages and disadvantages. First is the inoculated method. This method has a known starting population due to the spiking of a known species on the device, as well as a set of control samples. The inoculated method requires artificial contamination of the bioburden on surfaces of the device, which may not be representative of where the native contamination is dispersed on the device. This method also only measures the removal of a certain test organism. The native exhausted method utilizes the naturally occurring bioburden through the product's manufacturing process. Therefore, the starting population is unknown. Additionally, many products are manufactured in a cleanroom environment and have low bioburden. If the bioburden is extremely low, the data may be insufficient to determine the correction factor. Exhaustion, as defined in ISO 10993, is a 90% reduction in the population, or a 1 logarithm, from the first extraction to the last. If the number of rinses of the product method does not achieve exhaustion, the test may need to be repeated. Additionally, if there is potential for inhibition of microbes in the bioburden test, additional testing may be needed to screen for adverse substances and ensure those substances are neutralized for the enumeration of bioburden. ISO 11737-1 is the standard that describes the bioburden test and recovery efficiency testing, or test method validation. ISO 22456 is currently in development for the guidance on tissue-based product requirements. Revalidation of the test method should be evaluated on a scheduled basis, and anytime there is a change to the manufacturing process, materials, device, etc. And this evaluation should be documented. So let's carry out an example of the inoculated bioburden method validation. We start by using a specified volume of a liquid suspension of Bacillus species, targeting an approximately 200 spores to be delivered onto the surface of the product. We allow the liquid to dry on the product. We then place the product into a sterile jar with sterile extraction fluid, followed by sonication and mechanical shaking of the jar. We then assay the extraction fluid by filtration for colony-forming units and compare the product results against a set of control samples. We recover an average of 125 colony-forming units from the control samples and an average of 105 colony-forming units from our test samples. We calculate the recovery efficiency by dividing 105 by 125 and multiplying by 100 for 84% recovery. The inverse of this percentage is our correction factor, which is 1.2. This correction factor would be multiplied by routine test results to correct for the inefficiency of our method. If we recover 80 colony-forming units from the product in a routine test, we would multiply it by 1.2 to yield a calculated result of 96 CFU. From the native exhaustive bioburden method validation, we place the product in a jar and add sterile extraction fluid. We sonicate and mechanically shake the sample, then assay the extraction fluid by filtration. We then re-extract the sample in the same manner 3 additional times, filtering the extract from each rinse. We obtain CFU results from the 4 rinses and notice that we have achieved exhaustion as our first rinse is 130 CFU and our last rinse must be 13 or less, 1 log reduction. We take the first rinse and divide it by the total of all rinses and then multiply the result by 100. This gives us the recovery efficiency of 74%. Again, the inverse of our recovery efficiency is the correction factor, which is in this example would be 1.4. Typically in a sterilization validation, we perform comparative resistance at a sublethal exposure to the sterilant. Our goal here is to show that the product bioburden is less resistant to the sterilization process than the biological indicator organism used throughout the remainder of the validation study. This comparison ideally shows no growth of the product bioburden after the sterilant exposure, but some survival of the indicator organism. The test of sterility of the product is a qualitative test, meaning it yields a positive or growth, or negative, no growth result. We can perform the test of sterility by cutting up or disassembling the product aseptically in a laminar flow hood. The device is placed into a liquid growth media and incubated for a minimum of 14 days. Alternatively, the product could be extracted similar to a bioburden test, with the extract filtered and the filter placed into liquid media and incubated for a minimum of 14 days. Similarly, if the product is a liquid, It may be filtered and the filter immersed in the liquid growth media for a minimum of 14 days. If the extraction method is utilized, extraction efficiency and a risk assessment must be performed. The test of sterility is detailed in ISO 11737-2. Additionally, USP 71 might be referenced. For products from animal source materials or packaged in a manner that removes oxygen, additional media types may be necessary to ensure recovery of organisms that grow in the absence of oxygen. Some considerations for tests of sterility for product during comparative resistance testing in a sublethal cycle is mitigation of resistant or significant populations of organisms in difficult to sterilize locations. Non-sterile stopcocks have mated surfaces that can be difficult for gas penetration. Consideration of sterile stopcocks may aid in a successful study. It is important to communicate to your project manager or laboratory of known locations of the device that may not have gas penetration and do not have patient contact. For instance, if there are hypotubes, tubes within a tube in a shaft of a delivery catheter, where the testing lab may perform a cross-section cut, which would expose a likely non-sterile lumen or mechanical features, this can lend to a failure of the test of sterility. Components that could break down during immersion in test media during the test incubation of 14 days, such as batteries, circuit boards, etc., can also be problematic in this sterility test, as they can appear similar to growth in the test media, or they can inhibit growth. Device coatings may also break down during immersion in the test media, exposing bioburden under the coating. This also stresses the advantage of something like a 70% IPA cleaning of the device prior to application of any coatings. It may also be worthwhile to test the uncoated device for bioburden to better understand the potential for this type of issue. Large and complex devices create testing challenges, as extensive manipulation and handling increases the potential for incidental contamination. It is best to provide the testing laboratory with an assessment of the sample with an assessment sample to develop a plan for testing prior to sample submission. The sterility method suitability, also referred to as bacteriostasis fungistasis, is our test method validation for test of sterility. The intent of this test is to ensure the product does not inhibit growth in the liquid growth media, resulting in a falsely negative result. We prepare the samples in the same manner as the test of sterility. However, we inoculate the samples with low levels, less than 100 CFU, of specified test organisms. We utilize 3 organisms per media type. Control samples utilizing the same type of test container and media volume are inoculated with the test organism as well. The positive controls and samples are then checked for growth within the required time frame. If no growth is observed in the test samples the test will need to be modified and executed with additional samples. Again, the BNF test is to ensure the product does not inhibit growth in the test of sterility. We test each media used and specify the media type and the volume of media used. The other portion of showing comparative resistance would be the biological indicators that could be used for internal process challenge devices, or BIs, inside the product or product packaging, and external process challenge devices, BIs, on the outside of a pallet, in the fractional cycle, and also used in half and full cycles. Most commercially available biological indicators are cultured by immersion in 10 milliliters of tryptic soy broth, also known as soybean casein digest broth. Biological indicator organisms utilized for gas methods would be Bacillus atrophius 30 to 35 degree incubation, used for ethylene oxide sterilization, or Geobacillus start-thermophilus 55 to 60 C, often used for vaporous hydrogen peroxide sterilization. Standard spore strip disc biological indicators of both organisms are typically incubated for a minimum of 7 days. There are other types of biological indicators available commercially that require the use of manufacturer's media and may require other incubation temperatures and durations. Biological indicators may be tested as part of routine release of products through validated sterilization processes. Certain types of sterilization validation could utilize test of sterility, such as overkill method or fraction negative BI bioburden approach, or an enumeration of the biological indicator population through the BI bioburden approach, direct enumeration, after varying levels of sterilant exposure. ISO 11135 has more information on EO sterilization validation methodologies. ISO 22441 is currently under development for industrial vaporous hydrogen peroxide sterilization validation. ISO 14937 has guidance for validation of other sterilization methods. Biological indicators have a certified D-value from the manufacturer. D-value is the decimal reduction time and is the measure of the resistance of the organism under controlled conditions. The D-value is the time or dose required to inactivate 90% of the population of the test microorganism at controlled conditions. This D-value is specific to a specific species or strain of organism under the controlled conditions. So if we had a population of 1 million spores of a microorganism, the time and dose required to reduce the population to 100,000 colony forming units or spores would be the same time or dose required to reduce the population from 100,000 to 10,000, and the same time or dose to reduce the population from 1,000 to 100. If we had a starting population of 1 million CFU, or 10 to the 6 CFU, of a specified organism, and it took 8 minutes to reduce the population to 100 CFU, our D-value would be calculated by taking 8 minutes divided by the log base 10 of the starting population, which would be 6, minus the log base 10 of the final population of 100 CFU, or 10 to the 2 CFU, which is 2. So 8 divided by 4 gives us a D-value of 2 minutes. Medical devices target a sterility assurance level of 10 to the minus 6, which is the probability of 1 in 1 million of a non-sterile product. For ethylene oxide, the sterility assurance level is calculated by using the BI along with the population of the BI and the tests of sterility from the validation cycles. Again, the lower and less resistant the bioburden is from the product, the greater the likelihood of success in the fractional cycle. From this graph, we can see that as the D-value goes below the x-axis, we start to obtain negative exponents, indicating probabilities. So if our half cycle at 12 minutes of exposure has no growth from our biological indicators, we have reduced the population by 6 logs. If we double the exposure to 24 minutes, we now achieve the probability of 1 in 1 million, or 10 to the minus 6 for non-sterile product. Sterilization through certain gaseous methods has the potential to contribute residues to product that could have biological effects to a patient. Ethylene oxide residue testing and product residue requirements are detailed in ISO 10993-7. The ISO 10993 series covers other methods for biocompatibility testing as well. EO residue testing is critical to understanding the risk to patient safety. EO is known to exhibit several biological effects as an irritant, mutagen, carcinogen, and can have deleterious effects to organs and reproduction. We analyze ethylene oxide residues through gas chromatography. Gas chromatography involves an inert, unreactive gas such as helium, shown in the gas cylinder on the left, which acts as a mobile phase to move chemicals through the column, the circular item in the column oven. The mobile phase goes through traps which filter out water and hydrocarbons from contaminating the column. Sample extracts are injected into the inlet, where they are volatilized at high temperature. The chemicals move through the column with the mobile phase gas, and are separated due to differences in their reactivity to the stationary phase that lines the interior of the column. When the chemical reaches the detector, it is translated to a detector signal that shows as a peak on a data processing unit. The time it takes for the analyte to go from injection to detector is known as the retention time. NIST traceable reference standards of the chemicals of interest are analyzed at several dilutions. The standard curve is used to correlate the detector response, or area under the peak of the chromatogram, at the retention time of our analyte to give us the amount of the analyte in our unknown sample. Quantitative analytical chemistry methods will have limits for detection and quantitation. The practical quantitation limit is the limit of the method and instrument to accurately provide a measurement of the quantity of an analyte. Values below the limit of quantitation may indicate the presence of the analyte, but the accuracy of the measured amount is questionable. Method detection limit is the limit of the method and instrument to detect the presence or absence of the analyte with respect to the baseline signal. Ethylene chlorohydrin residues are obtained through reaction of ethylene oxide with chlorinated compounds, such as bleached paper. Ethylene glycol is formed by the reaction of ethylene oxide and water. Limits for ethylene glycol were removed from the ISO 10993-7 Annex. Annex 1 discusses its removal in detail. We typically test patient contact portions of a device for ethylene oxide and ethylene chlorohydrin residues. There are two main methods of extraction identified in ISO 10993-7. The simulated use method involves a single extraction of a device in water. A portion of the extract solution is used for injection into the gas chromatograph, and the amount of our residue is measured. The second method is an exhaustive extraction, which involves repetitive 24-hour extractions. The extract is analyzed after each 24-hour extraction to determine the amount of residue in the sample. Extractions continue until there is a 90% or 1 log reduction in the amount of the analyte from the first extraction to the last. The determination of whether a simulated use or exhaustive method of extraction is used is based on the intended use of the device. Ethylene oxide residual testing is normally performed during sterilization validation activities. Samples are pulled from the sterilization load after a minimum amount of aeration. The samples are frozen to prevent off-gassing during storage and shipping. A sample blank, which is non-processed, is tested to ensure no interfering chemicals in the product extraction are affecting the measurement of EO and ECH. Often multiple aeration time points are tested, as well as after both one full sterilization cycle, referred to as a 1X, and two sequential full sterilization cycles, 2X. 2X processing is performed to ensure reprocessing of a product sterilization run yields acceptable product residue levels. Some devices may require splitting portions into separate samples due to patient contact time. For instance, a stent with permanent patient contact requires exhaustive 24-hour extractions, while the delivery catheter may have limited patient contact, resulting in a shorter single extraction of the catheter. Some devices may require a fluid path, and others may be fully immersed in the test solution. The size of the device and the volume of the extraction fluid affect the detection of the test. The detection limit needs to be able to detect the acceptance limit for the device. The test container is filled entirely with water, as headspace or air can affect the stability of the sample. Extraction parameters are based on worst-case patient contact for time and, in some cases, temperature. For instance, if the medical procedure for the device normally has a 1 hour patient contact, but could in worst-case conditions take up to 3 hours, the device would need to be extracted at 3 hours. Extraction temperatures of 37 C, body temperature, are used for devices that are wholly or partially in contact with the body during use. Room temperature, 25 C, is used to extract devices with no immediate patient contact during use. An example would be hypodermic needles. Headspace analysis is performed within a closed vial with air at a 100 C. ISO 10993-7 details the limits for devices. The limits identified in the standard are based on a 70 kilogram adult. An amendment to ISO 10993-7 was issued in December of 2019 for guidance on establishing residue limits for special populations if applicable for the device, including premature neonates, infants, and children. Limited contact devices are those with cumulative, multiple or repeated use or contact up to 24 hours. These devices cannot exceed 4 milligrams of EO and 9 milligrams of ECH. Prolonged patient contacting devices are those that have use or contact that is likely to exceed 24 hours, but not more than 30 days. Prolonged patient contact devices cannot exceed 4 milligrams of EO and 9 milligrams of ECH in the first 24 hours, and cannot exceed 2 milligrams per day for average daily dose. Additional limits also apply of 60 milligrams of EO and 60 milligrams of ECH in the first 30 days. Permanent patient contacting devices are those whose use or contact exceeds 30 days. Permanent patient contact devices cannot exceed average daily dose of 0.1 milligrams per day of EO and 0.4 milligrams per day for ECH. The same limits for first 24 hours and first 30 days apply. Additionally, limits for 2.5 grams of EO within a lifetime and 10 grams of ECH in a lifetime apply. Tolerable contact limits apply to surface contacting devices and implants. The surface area of the patient contacting portion of the device is needed to perform the calculation. The purpose of the tolerable contact limit is to provide localized to prevent localized irritation from EO and ECH released from the device. A negligible irritation result from the skin sensitization test can also be used. The TCL for EO is 10 micrograms per centimeter squared, and ECH is 5 milligrams per centimeter squared. Additional device limits for additional device-specific limits can be found in ISO 10993-7 for items such as blood filters, drapes, intraocular devices, and others. As an added note, endotoxin testing is required for routine testing of devices contacting the circulatory system, cerebrospinal fluid, certain implants, intraocular devices, and devices with non-pyrogenic label claim. Current testing follows USP 85, USP 161, which is specific to infusion-transfusion devices, and ANSI ST 72 provides additional guidance.
Thank you all for attending today's STERIS Tech Talk.